Hi, everybody. My name is David Gregg. I'm the director of the Rhode Island Natural History Survey, and this is our third live stream. It's in the You're Never Too Old to Learn New Things department. Today, for a long time now, we've had Larry Millman come to Rhode Island and do mushroom walks and talk to us about books he's producing and things like that. So, Carrie, you need to mute that so it doesn't have feedback. So Larry is going to do a virtual mushroom walk, which I videoed yesterday and put up on our YouTube channel. And now we're trying a live stream where Larry's going to talk about some of the mushrooms that we found, some of the fungi that we found, and other interesting mycological things that this brings up. So I'm going to turn it over to Larry Millman, and was that all the things that I was going to do? Yeah. Kyra is monitoring the chat, and Larry has agreed to take questions. So if you want to, if you have questions, put them in the chat. If they're relevant to the discussion of the moment, Kyra will flag us down, and we can talk about those. But we'll have a Q&A session at the end. Yep.
Should I sit down or stand?
No, you can. I think you can stand. Take us a little bit to adjust the cameras, but nice to have everybody here. And so here's Larry Millman.
Thank you, David. Yes, I'm being decapitated.
There you go.
Thank you. Good. Thank you, David and Kyra, for hosting me. And this has been probably the driest season for in New England in living memory, and the result has been not so much a world bereft of fungi, because fungi, and by the way, what I should do is distinguish between fungus and mushroom. Typically, a mushroom is a mushroom-shaped mushroom, and a fungus is a non-mushroom-shaped fungus. But for purposes of this talk, which is being directed toward so-called citizen scientists, I'm just going to use the word fungus and mushroom informally, interchangeably. Anyway, I mentioned the drought and how there's been a paucity of mushrooms. And indeed, what we have here, which may look like a bounty to many of you, is for early August, beginning of July, somewhat of a paucity rather than a bounty. But what often happens during dry periods is that fungal parasites go into overdrive. Their hosts, the fungi that are indeed being attacked, don't have the appropriate defenses to defend themselves against them. Think of it as having compromised immune systems. They're not getting moisture, which they desperately need. Imagine a person in the Sahara Desert without water. Well, fungi are a little smarter. They simply do. Their mycelia, the vegetative part, doesn't send up mushrooms. The mushrooms are not, in other words, in the Sahara. They need the water. If they don't have it, when they fruit, and sometimes they fruit in very idiosyncratic shapes because not enough moisture, but sometimes they don't, in which case the parasites take over. Now, here is a very common one. It's a bolete that has been parasitized by what's called the hypomyces. And this is another one. This is a scleroderma puffball that has been also parasitized by a hypomyces. A bolete, by the way, is a Basidiomycete, which means it produces its spores to be dropped or blown by the wind, gravitropically. There's an alternative, and this will be what should we call it? A test for size with respect to this particular type of stream. These are cup fungi. They're ascomycetes. And a morel is an ascomycete, and typically they tend to shoot their spores up rather than drop them down. And the parasite on this Basidiomycete is an ascomycete. It's what's called a hypomyces, and I think it's Chrysanteron. But I'm going to try also not to bombard you with Latin binomials during this presentation.
Is that the orange that you're looking at on that?
Yes, it's the orange that I'm looking at. It starts out whitish on the bolete, and it covers the pore area. You could say the pore area, the fertile surface, is what the fungus regards as being its most important part. So imagine your most important part. I mean, you know, you would go into a state of absolute melancholy, and the equivalent with the fungus is a state of, well, gosh, what's the point? And then the hypomyces takes over. So now here's another one. And I've got to, I actually broke this one. It's Boletus Parasiticus. And it's a scleroderma puffball and a bolete. And for the longest time, it was thought that the scleroderma, the puffball, parasitized the bolete. No, we now know that it's the opposite. The bolete is doing the parasitizing of the puffball. And here's a small one that shows how they together, how they're joined. This is a very uncommon species, but one could argue that in periods of extreme dryness, it's less uncommon because, as I said, parasites go into overdrive during such periods. Now, lichens, in case you didn't know it, are members in good standing of kingdom fungi. The lichen is a combination of fungus and algae, or two fungi and an algae, or two fungi and a cyanobacteria, or two fungi as one fungus and a cyanobacteria. This is, they are known as extremophiles. This is not an extremophile. What that means is they can tolerate conditions that other members of kingdom fungi cannot tolerate. Extreme hot, extreme cold, extreme dryness, and they just desiccate and revive according to what the conditions are, and they can desiccate and remain that way for 10 or 15 years if they so desire. There are lichens in the Arctic that are several thousand years old, and of that period of time, they've been undesiccated perhaps for 100 years or so. Now, this happens to be a very, very common one. You'll see it on trees everywhere. It's not doing much damage at all to the tree. It's using the tree as a perch so that it can get its energy. The algae can photosynthesize. Typically, no fungus can photosynthesize. It's one reason why the fungus has joined hands with the algae to create the lichen. This species, which I'm going to now toss out a binomial, Flavoparmelia caparata, is among the most common of all lichens in the Northeast. And also, many lichens do not do well in urban habitats because of the pollution. One thing that happens is the pollution keeps the algae from photosynthesizing. It blocks the sun. This one, however, which I'm displaying for you, you'll see it everywhere on branches, fallen branches, trees, etc. Has no problem whatsoever. And it will be in dirty urban centers where automotive exhaust is the dominant chemical, or elsewhere. Flavoparmelia caparata. Now, I ought to say one other thing with respect to lichens. They are used in Europe for pollution monitoring. range of species in a particular habitat, indicating how polluted that habitat is. It would be nice if some of this was being done in the U.S., but the U.S. tends to be a somewhat primitive place with respect to ecological studies. We go on. Now, polypores. We found a few polypores. Here's one that is a perennial, and you can tell it's a perennial if you count, let's see, the tube layers. Each tube layer is a year of growth, and this is changing because of climate change, where if winters are less severe, they will continue growing. But typically in the past, there would be a sealing off of the tubes by the fungus to protect itself and its spores and its pores. Remember, as we said before, most important part, this would not exist. Every fungus you see is an evolutionary adaptation that is there to produce spores, not to aid and abet your palate, not even to aid and abet the biodiversity, but to create spores. Anyway, usually there are a lot of polypores even in dry seasons because wood holds moisture better than the ground. But in super dry periods, and our spring and early summer have been super dry, there are not very many polypores either. These are polypores that are soft. This one, as you can see, is hard. If I threw it against that screen, there might crack. It probably wouldn't be a good idea. But this was related to the cheese polypore, and they are cheesy and soft and so on, hence the name cheese polypore. A lot of polypores, in fact, most of them are not bracket-like. They are what's called resupinate, and they just grow supinely on the surface and produce their spores this way. A lot of them that are like this have not been studied because they're not obviously charismatic, they're not colorful, they're not edible, etc. But they're recyclers, and they're very, very good at that. And in this instance, the polypore is recycling. And again, it's the mycelium that's doing the recycling, not the actual fruiting body. This is what's called a white rotter. What does that mean? It is, mycelium is eating the lignin in the wood and leaving the cellulose. The lignin is what's responsible for the wood's rigidity, either in the upright stage or in the fallen stage. What's the result of having your, I don't mean you personally, your lignin eaten is that you become loose and cottony like this. See, that's an example of white rot that the mycelium is performing. Another one that is interesting, the aerial view is never enough when looking at a fungus. You have to look at the underside, and I don't know how visible this is. These pores of this species, it's related to a turkey tail, are what's called, they're irregular to the point of being dedaloid, D-E-A-D-A-L-O-I-D. And this refers to Daedalus, who created the maze or labyrinth in which the minotaur lives. But you needn't worry about finding a minotaur inside these miniature mazes. And it's maze-like, hence the name Daedaloid. And that is a way of identifying certain species of polypores where, unlike this, for instance, which I just showed you, these are very small, round, super round pores. Hard to see in this case, but bring a hand lens to the field and you will indeed be able to look closely at the pores of polypores. We will now move on to Basidiomycetes and fleshy fungi, mushroom-shaped mushrooms. And what happens there are certain types of mushrooms that tend, in a very dry period, to, I wouldn't say thrive, but they show up and they make people somewhat annoyed because they seem to be keeping out the ones that are more interesting. And I'm referring to Russellas. These are Russellas, and we saw a great variety. Say we saw 10 to 12 species on our walks the last 2 days, and they have developed the ability to fruit during difficult times and also during cold times. So you tend to see Russellas when you often don't see anything else. Russella suggests red, and 50% of all Russellas don't have red caps. They have yellow, green, purple caps. And one of the things about Russellas, they're hard to identify, but there are ways to identify. And first, identifying it as to a Russella, they're called Brittle Gills. And if you do like this, the gills will just simply fall out, and in this case, make a mess of the table below. Also, you've got a Russella and you want to identify it. One of the ways, and you need to get a good field guide too for this, is the cuticle peels and how far that cuticle peels will help you identify it. In this instance, I'm peeling it, and it is heading, let's say, not very far, maybe one third of the way toward the center. That would be a diagnostic feature, along with the color, and then typically with Russellas you need a microscope to identify them. So we now move on. Russellas are in the same family as milk caps, and this is a milk cap, so named because if you disturb the underside, and I think you might be able to see the little dots of milk coming out. And by the way, this isn't real milk. It's not something that you would use to substitute for a milky drink. It's called latex, but it's not really latex either. It is colored water released by the mushroom when it's injured. And that color often is a way of identifying a milkcap or lactarius. Also, it oxidizes. So if it's yellow or white at the beginning, wait a little while, it might turn yellow or green, in which case then you might find you've got a leg up on identifying it. Here are a couple of different species. These little orange mushrooms, if you can look at them closely, and again, it may not be possible, so you're going to have to take my word for it. These are miniature chanterelles, but they're actually miniature chanterelles, Cantarellus minor. They're not full-grown. Well, they're not the kind of chanterelle that you would want to collect for the table, because they're not going to get much bigger than this. But it's important to look at the chanterelle and see that they have ridges rather than gills. That's important for distinguishing between them and certain other mushrooms like this one, which is called the hygrossidae, which has gills and not ridges. The ridges, if you look at them, they don't go too deep into the interior. They're more surface, whereas these go deep into the interior. About this time of year, boletes start to appear. And we found a certain variety of, well, one of the species that was common is Boletis Bicolor, so named because it is reddish and yellowish. But it should be called Boletus Tricolor, because when you put your finger across the pore surface, it will turn green again, this is an oxidation reaction. And with respect to some of the species, you can identify them based upon how long it takes. Some of them will turn green after you bruise them. It might take 3 or 4 minutes. And some of them will take right away. And I should say that due to phylogenetics and DNA studying, Boletis Bicolor is really a species complex. This is one of 10 different species that are called Boletis Bicolor. And, as time changes and new books come out, it won't be bicolor, it'll be something else, tricolor, monocolor, or something else like that. We found a couple of other boletes too, and some of these will stain. Actually, these are a little old, but staining brown is not uncommon over the pore surface. And again, the bolete has pores, so it shan't be confused with, say, a Russella, which has gills. And also, they tend to be very fleshy. The context, which is the area between the surface and the cap, so fleshy that they become popular condominiums for insects. And the insects in question, in fact, have it good because they don't have to worry about finding food anywhere else. They're eating their condo, and so they don't have to leave home. They are happily quarantined inside the context of the bolete. We go on now to Amanedas, and this sometimes causes a cringe of terror when you mention the word Amaneda. There are some toxic Amanedas, but even at the most toxic, and this isn't one of them. This actually is one of the edible ones, Amaneda fulva. People say, oh, if I touch it, do I need to go and wash my hands? Otherwise, will I die? And the answer is no. You need to get a chunk of it in your stomach in order to die, if that's what you want. And even then, if you go to the hospital, to the emergency room, there's a good chance you won't die. No, having this, I can put this all over my mouth and no problem. But if you do touch someone else's computer keyboard, especially now, you better go and wash your hands. Not only various bacteria that are alien to you, but certain viruses might be on those keys. And same with somebody's cell phone.
So how do you know that's an Amaneda?
How do I know this is an Amaneda? Instinct. It's not here because it didn't survive the transportation. It had a bulb at the bottom, like this. This is another one. This is called the cleft foot.
That's just the bottom part.
That's the bottom part. And with Amanedas, if you have an instinct that it's Amaneda, you want to try to get all of it out. You don't just sort of cut it off by the stem, but you want to get it all out of the ground so that you can identify not just the cap, not just the stem, but the bulb at the bottom as well. That is a way of doing it. Also, the warts here, which I think you can see on the cap, these are the remains of the universal veil that would have covered the youthful Amaneda. And as it grew, the cap would expand from being called a button, breaking the veil into little warty tidbits like this. And in most cases, this is an unusual Amaneda because most of it have veils or rings. Like, for instance, let's see, right here. This is what's called a pendant veil. You see this right here with this Amaneda. And that is the flesh that connected the cap to the stem. And as the cap is expanding like this, the tissue breaks and clings to the stem. And it has been suggested that one reason they've evolved to do this, not this one of course, but the one I just showed you, is that this helps protect the fertile surface from insects that are coming up and might want to eat it. And again, actually they have done some eating with this one. There's what's called the Red Queen hypothesis. Let's say an organism develops an ability to fight off a parasite or a diner. Then the diner develops something that will triumph over the host development, and then the host development something else, and so on and so forth. And, you know, one imagines in situations like this where insects have been. And once again, with respect to the toxins in Amanedas, one of the suggestions has been they're there to keep insects away, not to kill Homo sapiens, but to keep insects away. They evolved well before Homo sapiens, and it appears they will survive long after Homo sapiens. So here are a few Amanedas. This is one that will, if you had cut off the stem, you wouldn't realize it has what's called eradicating root right here. And that is indeed how I was able to identify this as Amaneda onusta. Not a very common species, but again, so many collectors are unaware of the importance with Amanedas of collecting all of it, that maybe they just would collect this and not identify it. And this is, one can easily guess this species based on the color. It's called flavorubescens, flavo-yellow, rubescens red. And you see the stem, the ring has, again, in transport, disappeared with a red cap, and this red hump is called an umbo, and that again is a diagnostic feature for identifying certain mushrooms. By the way, I should mention that my most recent book, Fungipedia, it's not a guidebook. It's doing something rather different from what I'm doing here. It's giving you a broad context that includes the history of mycology, the ethnographic ways in which fungi have been used. And one such way that I document in the book is where a puffball, and I don't have any of the other puffballs, is used as a styptic. You cut it open. When it's sporulating, put it on your bloody injury, and it will cause the blood to cease to flow. Also, they have antibiotic properties. Fungi and bacteria are common enemies, and they are commonly trying to fight off bacteria, and they tend to do a good job. Putting those spores in your wound has an antibiotic purpose, and it helps. Native people in the North and in Central America have commonly done this with puffballs, and it's been somewhat successful. Anyway, I shall now go on, get away from being mercenary. And I did mention, this is a Amaneda, it's called the Cleft Foot. And again, it's, transporting, I particularly like a lucite container, but I don't know one that's quite big enough for a species with this long a stipe. So the Cleft Foot Amaneda, alas, the foot got amputated and the head got decapitated during the journey. I have mentioned ascomycetes. We didn't find too many today, The ones that you do see are blackish. They're called Perennomycetes. But in the spring, not this spring, which was lousy for any fungi, typically they tend to grow because they are not overly competitive. You know, a Basidiomycete like this bolete has mycelia that are far more aggressive than the mycelia in this little cup fungus. So when the season comes for this tends to lay low. And fungi, by the way, are varying degrees of competitive. In a log, you'll have any number of species competing for the food. And some of them are being so outcompeted that they only remain mycelia. They're not producing fruiting bodies. Carl Zimmer, whom you may have heard of, the naturalist, did a survey of a 6-foot log and found 298 species of fungi inside of them. And there were probably only 2 or 3 fruiting bodies. So onward, here is another Basidiomycete, and it's called the coral mushroom. And this one is one of the rare ones that grows on wood. It's it used to be Clavocorona pixidata, but the genus name, like all genus names, are changing, and one sometimes feels if one's suffering from early onset Alzheimer's because of all these changes. But one can easily identify this one because it grows on wood. It used to be Clavocorona pixidata. Common name was pixie coral. And the substrate with this, as with so many fungi, helps you determine what it is, because there ain't too many corals that grow on wood. Also, when I said Basidiomycete, this doesn't have gills, doesn't have pores, and it doesn't have teeth. So where are the spores being produced? They're being produced in the upper part of the coral segments. And you know, it's the basidia which are producing the spores are there, invisible to the naked eye. You can't see them, but they can see you. And you know, constantly are those spores coming out. In the course of a lifetime, this would produce probably, let us say, 100,000, 200,000 spores. In the course of a lifetime, this polypore might produce 10 billion spores. It has a longer life expectancy than that coral fungus. By the way, I should mention this. This is a work of art that I created. And I wanted to find out if a particular species was a pluteus, a deer mushroom. When you're doing a spore, you do a spore print because the underside of the mushroom may not be the same color as the color of the spores. And the color of the spores will help you identify the mushroom. How do you resolve this dilemma? You take the mushroom and you remove the cap like that, and then put it down on a piece of paper, white or black. If you think the spores are going to be light colored, black paper would be good, especially if you're thinking of creating a work of art. And then make certain you put a cup over that so the specimen will not dry out before it drops its spores. And this is the result after about 6 hours. These are pinkish spores which confirm that the specimen was a pluteus, common pluteus, Pluteus cervinus, otherwise called the deer mushroom, D-E-E-R, not D-E-A-R. And I recommend this to you both because it's aesthetically appealing and it helps with fungal identification. Now, we're getting more toward the end. What's left here is a few others. Now, this is a species that has a very irregular underside. And I recommend everyone gets one of these. This is called a hand lens or a loop. And you can get them online for $5 or so. And it's important because you can then look at this and see that these are sort of ridges and they're sort of cross-veined.
What magnification is that?
This is 10 power. That's
the right amount.
The right amount, but 15 is good too. And so this, I think, is what's called a Paxillus. I first looked at it without the hand lens and thought, oh, it's another one of these mini chanterelles. But looking at the surface, I saw that it wasn't. Now, with this, I will need to do a look at the spores. It's, I think, an inocybe species, INOCYDE. And these are very interesting. You know, this is a little dry, so it won't have an odor, but their odors range from sort of bleach-like to sweet corn to spermatic. And that helps you with the ID. But what's really interesting are the shape of the spores. If you look at them under the microscope, 1000 power. They can be angular or trapezoid. And, you know, that in itself, even more than, say, identifying the mushroom, could be something that would bring you a sense of astonishment. It's one of either 2 or 3 inocypes, and I'll be able to determine which one once I put it under the magnifying glass. By the way, I forgot to show you this polypore. You know, it was created not by a mycelium, but by an artist named Francis Topping. And this is a very common one that I'm surprised we didn't see today or yesterday. It's Ganoderma applanatum, otherwise called the artist's conch. And it's called the artist's conch because people love to take the underside and draw cute little houses, cute little animals, and so on it. And in certain places like Labrador, they will sell this as souvenirs, a picture of a cabin near a lake. Well, I'm mentioning this not as another example of art, but as an example of human ignorance. When I was last in Labrador, I saw dozens of these in shops. I didn't see a single one on a tree. These have evolved with the trees there, and now that most of them have been removed, the ecosystem will be quite different. And personally, I think this species is far more artistic when growing on a tree or being displayed in a work of art like this than in a souvenir shop. Its underside has been drawn upon by Homo sapiens. So that, I think, is the end of this presentation. Again, I eagerly recommend for you to buy this book. I eagerly recommend that the Rhode Island Natural History Survey become your home away from home, and you go into the field and document both unusual and common species and share what you document with the Natural History Survey. In any event, now we will open this to...
Before we do that, I just wanted to let me come around here. So I wanted to mention briefly where we went, because it leads to my first question. So we went to 2 different preserves. We went to the Benson Preserve, which is the property of the Narrow River Land Trust, and thank you very much to them for hosting us at that preserve. And we also went to the Crawley Preserve, which is the work of the South Kingstown Land Trust and the Richmond Rural Preservation Land Trust. And that was also very interesting, and thank you to them. And so what I should say is that at the Benson Preserve, which is on the eastern shore of the Narrow River, we found virtually every mushroom you saw today was from the Benson Preserve. When we went to the Crawley Preserve, we found 7 or 8 mushrooms total. This one that Larry showed earlier is one of them, but was one of them. So my question is, what do you see, Larry, as the difference in the habitat between the Benson Preserve and the Crawley Preserve that made such a remarkable difference in the fungi that we found?
Well, this is a very good question, and it's unfortunate that so many people who are trained mycologists don't look at the broader picture and think of the ecology of fungi, where they're growing and where they're not growing. With respect to the Crawley Preserve, there was considerable damage by tent caterpillars of the trees. We did not see too much in the way of gypsy moth. And we didn't see too many trees standing high and tall. There was an incredible amount of understory, and a lot of it was ferns and a lot of it other was plants. And the issue is that with no trees around, there are fewer and fewer mycorrhizal fungi. And mycorrhizal fungi are fungi like these boletes, and this Russella, and the Amanedas, which grow with the healthy tree and exchange nutrients with them. With an unhealthy tree, you might find a few parasites, and then when it becomes a log, there'll be recyclers. But when it's extremely dry, a lot of the wood just crumbled in our hands. And this would not have provided nutrients for fungi at all, for the mycelium. Also, when we were at the Crawley Preserve, we went high upland. And in a dry period, upland is not nearly as good for fungi as lowland, because what happens when there is any rain, it tends to filter downhill. And that's the reason. The other place hadn't been assaulted by caterpillars, tent caterpillars, and it also had a lot of fallen wood. It had a lot of debris, and a lot of what we found was on the trail. And trails are good places for certain mushrooms. I showed you this inosopy. This typically grows on trails, on shoulders, not in the heart of the woods, but in a sort of disturbed area. Disturbed areas are, in fact, excellent for fungi, which are opportunists. You would find more species along a trail in eastern New England than an old-growth spruce forest in northern Vermont. So I think that's the reason. Plus, humans are great taxiers of spores, and a lot of people go through these woods bringing spores, but the ones that would be taxing spores through the Crawley woods, the spores would fall off, and then where to go, what to do, nada. Whereas here, there are a number of still older and healthier trees that, with luck, the spores would fuse and form a mycelium, and there you are.
Okay. And Kyra, go ahead.
So we have a question from Emily and Brian. Can you talk a little more about the parasitic bolete? How has its life cycle ended up so different from that of typical boletes?
How has its life cycle ended up? This is very interesting. At a certain point in the distant past, it realized that, hey, I could probably not have to worry about giving nutrients to a symbiotic partner, nutrients being phosphorus, potassium, nitrogen, uptake of water. I can just find something else out there, parasitize it, and there's no exchange whatsoever. You know, I don't have to be cooperative. I can be parasitic. And at some point, it decided that's what it wanted to be. But interestingly, because this is called the pigskin poison puffball, and I'm going to break it open, and you'll see the inside. This is a little immature. It goes from grayish to dark grayish to blackish. Because of that, because that suggests something rather nasty, it is indeed toxic, so don't eat it. It was long thought that it was the puffball that was parasitizing the bolete. And only in the last, I don't know, 20, 25 years has it been determined that the bolete is parasitizing the puffball. And the bolete has done something that typically boletes don't do. They're mycorrhizal and not parasitic. But at some point deep back in its history, it realized that it didn't have to make all the cooperative adjustments that other boletes had. And it also could maintain its morphology. You know, it has little scales around on the stem. And if you didn't know, if you didn't find it with the scleroderma, you might not guess it was a parasitic bolete. It's called, the name is actually, it used to be Boletus Parasiticus. It's now Pseudo-Boletus Parasiticus, as if to say, well, this really isn't a bolete in the strictest sense. Any other questions?
Let's see. Just a comment from Elise Torello, who was one of our members. She said she saw a gorgeous Berkeley's Polypore and will send photos in a bit.
Berkeley's Polypore. I was telling David about this yesterday. This has been an extraordinary year for them. It is the largest polypore in eastern North America. It is spatulate, meaning it forms these spatuloid extensions. It could be mistaken for chicken of the woods or hen of the woods, but it's much bigger and it's whitish, creamy white in color, and can be 4 feet in diameter. And if you get my book, Fascinating Fungi of New England, you can see for scale its size, because there's a picture of me almost dwarfed by a Berkeley's Polypore. But for whatever the reason, this has been the best year for them I can recall. They grow with oak trees, and they grow at the base of the oak tree. And they're not a serious parasite. They're a parasite that takes advantage of an already weakened tree. And there could be a lot of reasons that going on. That the trees have been weakened through the drought. It could be that the last couple of years haven't been good years for Berkeley's Polypore, and now the mycelium is fully strong and going into the mode that it would always have wanted to, but, you know, and it's now doing. You know, there are a lot of questions with respect to kingdom fungi, and one of them is why this has been such a good year for Berkeley's Polypore. Another, you know, anyone who tells you that they can identify any fungus they see on a walk is a bald-faced liar. If I can get 75%, I feel I'm doing well. There were quite a few on our walks. This is one of them. Reddish stipe and intervenose gills. I don't have a clue. So I'm going to take it home, study it, look it under the microscope, see what my books say, and I might, in fact, utterly strike out.
There was a brown one yesterday that you said was maybe a rarer one.
It was this one.
That's
the anosity, yeah.
So then another release that she's been seeing lots of black staining polypore as well.
Oh, very good. I've seen only 1 or 2 this year. And that's good to hear. Anything else?
Nothing else from here right now.
Okay. Well, Larry, thank you very much.
Thank you, David. And thank you, Kyra. This has been
Pleasure.
great fun.
Thank you very much, Larry, and thanks to everybody for tuning in. We will continue to experiment with this new technology. We'll get better microphones, better cameras, better Wi-Fi signal. If you want to know more about the context that these came from, we did post a really rough cut of our mushroom walk from yesterday on the YouTube channel, so you can watch that. I took some more footage this morning. Might be a little while before I have a chance to upload that, but I'll try to cut that together and upload it. And we will be trying to think of new programs that we can do in this way, this virtual way going forward. So if you have any ideas or suggestions, we'd welcome those. You can email us. Yeah, thanks very much. And we will see you out there.